This video is going to be an introduction to what inflammasomes are. So right here I have a drawing of the inflammasome, and what it is it just consists of certain nod-like receptors that assemble with proteins such as proteases like ASC, which stands for, it's just an adapter protein, and it binds to the NLRP3, which is this certain type of inflammasome that we're going to talk about via the homotypic domain PYD. And then it also consists of caspase 1. So the inflammasome it was discovered by a guy named Jurg Chope and his colleagues, and it's just a large protein complex that activates caspase 1 right here to generate interleukin 1, which will then go to the outside of the cell, and interleukin 18 as well, so I should write that down. Interleukin 18 as well, and then they go to the outside of the cell, which generate pro-inflammatory cytokines. The NLRP inflammasome, which is expressed by different cells such as monocytes, macrophages, neutrophils, and dendritic cells, and then also some lymphocytes and epithelial cells. What it does is it responds to DAMPs and PAMPs, which are damage-associated molecular patterns, pathogen-associated molecular patterns, also ATP influx and crystalline influx. So there are kind of these three different models of NLRP3 activation, which involve this ATP triggering pore formation, lysosomal rupture by crystalline or particulate substances, and then reactive oxygen species signaling. So none of these are mutually exclusive, but they have all been proposed by different scientists. What inflammasomes do is they act in tandem with other pathogen recognition receptors in response to the PAMPs and the DAMPs to generate mature interleukin 1 and interleukin 18 via the caspase 1 mediated cleavage in order to prime the inflammatory response. So what the role of the inflammasome is to prime the inflammatory response by activating pro-inflammatory species.